Okay, so the serine protease that we're going to talk about here is chymotrypsin. So chymotrypsin is basically like a protease, or is a protease. It's in small intestines, and the way that it works is it's part of bile secretions, and what happens is the pH in your small intestine, which is about 8 at that point, a lot of the peptides from food, from protein that you eat, still needs to be broken down. And so what a serine protease does, in this case chymotrypsin does, is it's going to go in and it's going to use that serine as a nucleophile to break down peptide bonds in your food. Chymotrypsin is a pretty big protein, and it has, in fact, 28 serines. And only one of those serines is a nucleophile. Only one of those serines does the chemistry. So what makes serine 195 so special, that it can act as this nucleophilic catalyst in contrast to the other serines in the enzyme? Well, it's a couple of things. Number one, of course, is that it's part of that active site that binds the substrate. And number two is that there's other amino acids in the active site that are going to make serine 195 act like a better nucleophile. And I want to just mention here, to remind you, is that an alcohol is going to be a better nucleophile. This kind of primary alcohol especially is going to be a better primary alcohol, or better nucleophile than water. And the reason for this is that you get that alpha effect from the carbon next to the oxygen group there. So if you look at your periodic table at home, you can see that carbon is more electronegative than a proton, and that's going to increase the nucleophilicity. It's going to increase the electron density that is on that oxygen to have it act as a nucleophile. So chymotrypsin works by a pretty common process, and that is a catalytic triad. So what's going to happen here is that we have our serine 195 here, and basically what's happening is that the serine is next to a histidine, which is next to an aspartate. So this is the catalytic triad. So here's the nucleophile, and that's that serine 195. And these two, this histidine and this aspartate, are going to act to make this serine 195 act more like an alkoxide ion. So remember that the pKa of a primary alcohol is somewhere around, let's say, 16. And that means that you need a pretty strong base, and you can't have any water around if you want to deprotonate that alcohol. But in an enzyme active site, a lot of times you can make this alcohol act more like an alkoxide ion just based on how you precisely set up the spacing between these acidic and basic amino acids. And so in this case, right, we have the conjugate base of aspartic acid, aspartate, is deprotonating the histidine here, and that's this pulling electrons this way, and that's pulling electrons that way, and so we're deprotonating the serine here. So the way that the mechanism of chymotrypsin works is that in this case we have that serine nucleophile. So you could think of this already as kind of set up as a triad. So this is already like an alkoxide. So I would say this is probably already kind of deprotonated at this step, frankly. And what happens there is that that alkoxide-like anion is going to attack the carbonyl and the peptide of a protein. And then we're going to go through a tetrahedral intermediate where the alkoxide is now going to be formally completely deprotonated. And now when the tetrahedral intermediate collapses, we can also protonate the peptide NH to get a leaving group, peptide leaving group. And then we can hydrolyze. This is an acyl protein. We'll talk more about that in a second. We can hydrolyze that through enzymatic means, and then collapse that second tetrahedral intermediate and do some little proton bobbles here. Lose the acyl enzyme, and so now we've split that protein into two halves. So overall, this is the mechanism that it's going through, right? And so this, the previous slide, was really looking at it from more of the substrate viewpoint. But now let's also look at the catalytic cycle for the enzyme. So we're going to start here. So we have this catalytic triad set up. And so we're thinking more from the enzyme point of view. We're bringing in the substrate, and so here's what I was talking about before, how I kind of like to visualize it, is I kind of like to think of this more as pictures than formal arrow pushing in a lot of these cases. And so we have a tetrahedral intermediate, and notice that we have this oxyanion. So this oxyanion is actually stabilized by the enzyme. So the enzyme has a structure, a substructure in the active site that's able to stabilize the charge buildup for a tetrahedral intermediate in this reaction, and that's known as an oxyanion hole. And that's a common feature of enzymes that go through these kind of tetrahedral intermediates, is that they have aspects of their active site that are going to stabilize this oxyanion hole in the transition state. So in the next step, we're going to get here to this acyl enzyme. Essentially, we've broken the bond, get an acyl enzyme. It's acylated. We're going to hydrolyze that. Again, notice that we're still using that catalytic triad to hydrolyze that. It's not just a normal ester hydrolysis reaction. We're again stabilizing the tetrahedral intermediate. And at the end, we're going to get two halves of the protein. So this is overall looking at this oxyanion hole more in detail. What we have here is we can see how the enzyme does it. And so look, we have a couple of glycines. So remember, glycines are amino acids with an unusual degree of freedom. They're very wiggly. And these glycines have their amide NHs set up so that they can H bond really nicely with that minus charge in the tetrahedral intermediate that builds up. And so this is what's stabilizing the tetrahedral intermediate, is this little loop of a couple of glycines from the enzyme. So again, this is going to build up again. Just going to go through this kind of quickly, but again, we're getting that second oxyanion hole. And now we're going to have our substrate leaving. And now I want to talk about another example of this. And so this is the catalytic triad and subdolacin. And so subdolacin is a serine protease, but it's not related to chymotrypsin at all. So right, chymotrypsin is like a mammalian protease. This one is from Bacillus subtilis, which is can be pathogenic bacteria, but it also can live on your skin. But this is a Bacillus protease, and so this protease is totally this species is totally different than us. We're pretty different than a bacteria, but it's still the protease, which is kind of a bacterial defense mechanism. It still works by the same catalytic triad. So there's still a catalytic triad that's going to make this happen. And this is going to have our alkoxide ion, that charge build up there. And so they've done some really interesting mutational studies on this. So what they're going to do is what they've done is they've taken these pieces of the catalytic triad and they've cut them out, and substituted them with alanine. So that's the plain vanilla amino acid. So they cut them out and then seen how they affect the rate of the reaction. Okay, so normally we have a rate of the reaction. So here that's that kcat per second of the wild type. And then we can see versus uncatalyzed, so it's down 10 to the 10th versus uncatalyzed, right? So if we just take out that serine and mutate it to an alanine, then we reduce the rate by 1,000,000-fold already. So we've already changed the rate by 1,000,000-fold just by mutating out that serine. But also notice that if we mutate out that histidine there, we also change that rate, and we change the rate again by about 1,000,000-fold. And even if we mutate out that aspartate, so we're only switching out, that serine's still here in this case. The serine's still here, the aspartate's still here, we just got rid of the histidine. And so now all of a sudden we've completely destroyed the rate of this reaction by removing the histidine, but the serine nucleophile is still hanging around. But it's just a much crappier nucleophile now. It's the same with if we still have the serine and the histidine hanging around, but we got rid of that aspartate and we still destroyed the rate. And so you can see that that catalytic triad is really important. And if we get rid of all three, we're destroying the rate about the same as if we had gotten rid of any one. So you need all three of them there. So when you're trying to understand the mechanism of these nucleophiles, one of the things that you can do is use irreversible inhibitors. We'll talk more about inhibitors in a subsequent lecture. But one of the ways that you can get rid of one of these inhibitors is to have a nucleophile, or rather have something that's going to react with a nucleophile. So in this case, if it's a serine nucleophile, right, we said that that nucleophile is an alkoxide ion, right, or is basically an oxyanion, pretty close to it, something that's kind of, we could approximate it as that. And so one of the things about periodic trends, etc., is that phosphorus really likes oxygen. Phosphorus loves oxygen, and this has to do with how the orbitals between phosphorus and oxygen overlap. So phosphorus can also act as an electrophile. You know, it can act as a nucleophile, but when it's phosphorus 5, it likes to act as an electrophile. Phosphorus goes in between 3 and 5. And so what we can use is a phosphofluoridate. So in this case, we're using diisopropyl phosphofluoridate. And what's going to happen is that this alkoxide nucleophile can react with that phosphorus because it just loves the phosphorus so much. And what's going to happen is the fluoride kind of doesn't care to be there that much. And so at the end of the day, you're going to form a phosphate ester and a fluoride leaving group. And that phosphate ester, right, that's not nucleophilic anymore. A phosphate ester is not a nucleophile. And so now we have destroyed the activity of this serine protease. And as a note, and we'll talk more about this a little bit when we talk about enzyme inhibitors, this is actually how a number of, well, all of those dangerous neurotoxins that you hear talked about in chemical warfare. This is all how they work, is they're deactivating key alkoxide nucleophiles in different enzymes, and that's because this reaction is really fantastic in terms of how quick and effective it is. We'll talk more about that later. So here's another example of a nucleophilic catalyst here, and that is protein tyrosine phosphatases. In this case, we have a cysteine nucleophile, and then that's going to be acting as the nucleophile for this catalysis. And later on, or not later on, sorry, kind of zooming out, we can see all the wonderful ways that this protein is stabilizing the active site for this process. And so in this case, this is a tyrosine on the substrate is phosphorylated. We're trying to take the phosphate back off. This is a common way that cells send messages. And so this is happening in your body. All right, and with that, do watch the videos for the blink slides, and you'll see my arrow pushing. Yeah, I'll see you again on Thursday.